All right. Hello, everyone. My name is Hovanness Dingilian. I am a third-year environmental engineering major here at UCR. Currently, I am part of the Digital Agricultural Fellowship. I am currently working with my PI, Dr. Alexander Putman of the Department of Microbiology and Plant Pathology. And my project is titled Detecting Root Knot Nematode Damage Through the Use of Remote Sensing. So, when it comes to plants, there are many diseases and a lot of damage that happens. But the thing is there are very different causes. Some are fungi, some are bacteria, some are viruses, and others can be caused by another group, the unsegmented roundworms. And so that is what our focus is going to be for this project. And we are going to look at a specific one, which is the root knot nematode. Now, the root knot nematode is one of the three most destructive plant parasites in the world. You can see a picture here. They are extremely small. And the way that we are able to identify that they may be in an agricultural field or in a garden is because of the knots, as you can see on the right, that they leave in the roots of many crops, tomatoes, carrots, you name it, they can try and affect it. So the root knot nematode has below ground symptoms and it also has above ground symptoms. The below ground symptoms, which help identify that it is in fact the root knot nematode, is referred to as root galling. As we can see here on this carrot on the bottom, there are what appear to be even smaller carrots appearing. That is actually the root galls caused by the root knot nematode. What a root gall is that the root knot nematode burrows into the root and basically sets up a home for itself. And that's where it acts like a parasite and it basically takes the nutrients away from the plant. Moving on, we also see the above ground symptoms of the root knot nematode. These would include leaf chlorosis, which is the yellowing of plants, particularly the leaves. We also have stunting of the plants. The plants no longer grow to a height or width that is what is expected. And we finally have premature wilting. Basically, because the root knot nematode is taking the nutrients away, we start to see the leaves start to shrivel up, and that is wilting. One thing to keep in mind, though, is that these symptoms do not differentiate from other diseases. It's common among many diseases. And another symptom that also occurs is that it forms patches. These patches tend to be where the nematode is. It is called field distribution. And you're going to see one plant fully working, completely healthy. Then you're going to see another plant that may be affected completely by the root knot nematode. As I stated earlier, these symptoms are generic, though. So that makes it a difficulty to figure out if you actually have root knot nematode in, let's say, the agricultural field or the garden. So because we know that nematode damage is difficult, one of the things that also makes it difficult is that it starts below ground. And if it starts below ground, you may not notice until maybe 3 weeks after the root knot nematode has already been there. And so also the damage is very difficult to detect by the human eye at an early stage. This leads to our objective. So the objective of the study is to determine if remote sensing can be used to detect damage caused by the parasite root knot nematode at an early stage. If we're able to determine that there is the root knot nematode at an earlier stage by looking at trends, we may be able to save the crop. Granted, there may be some damage, but at least the crop would survive. So what can be done? Well, as we look at the root knot nematode, we will have to detect it early enough, correct? So we could potentially send a lot of people, researchers, into the into the field and have them look at every single plant. Problem with that is that it's not very efficient. It's very time consuming. So we will try another approach. One potential approach would be remote aerial sensing. Even if the nematode itself cannot be sensed, the damage from the nematode can be sensed. So what are some necessary steps for remote aerial detection? We could first develop drone flying and maneuvering knowledge. There can also be, after we've developed that, we can retrieve the data from instruments placed on the drone. And finally, we can analyze and interpret data from the instruments aboard the drone. I'll get into a bit more detail for this. First, the drone and instrument that will be used in this project. We have the drone, which is the AG bot by Aerial Technology Incorporated. And we also have the multispectral camera, or the MicaSense Altum by AG Eagle Aerial Systems, which will be used to take the images. So the data to be retrieved from the instrument on board the drone. Okay, how is our data going to come from the drone? It's going to come in the form of images, and they are going to have different color gradients, such as the ones we see below. These pictures are actually all the same field, except the only difference is they have a different spectrum. And here is the spectrum that I was referring to. For instance, the MicaSense Altum comes with 6 types of light spectra. One of them is actually not in the visible spectrum. And these images will all display different colors, but the important part is that they are all different wavelengths. So in order to find a trend, the individual pictures are going to be taken by the camera, and then we have to combine it together, which is called stitching. You stitch it together using a program, which then will give you an overall picture, which is referred to as a composite. And finally, the data will be interpreted by comparing the spectra with, let's say, a healthy field that maybe does not have any root knot nematode. Well, let's say you look at the root knot nematode and then you look at the field next to it that has the root knot nematode, then you're going to notice there's damage here, there's damage here, there's damage here. And these will be seen with the different color trends. So what are the future plans and research? Well, in the near future, I will be flying a drone over fields that will have planted processing tomato and carrot varieties. And these fields have already been identified to have root knot nematode. We will then do a process called the ground truth, which basically would allow for me to go to the field, and also our collaborators are working on this, where basically we would go to the field and we would check, is what we have on the map, is it correlating with the what's actually happening? And they would have something called the nematode severity data, which basically is, it will help determine how closely the remote sensing data matches the actual field damage. So factors that will be recorded that affect the drone may include something like weather. If you are shining a wavelength through the camera onto the field, you need a lot of sunlight. You know, if it's raining, if it's overcast, it's very difficult to actually get a good image. Something else that may make a difference is the development of the damage. If the damage, let's say, has occurred for a while, and we're already starting to notice the plant is starting to die, well, then this may be a good time to take an image. Or if we're starting to notice new damage on other plants, it might be a very good time to take an image. And finally, the key moments in the season. Tomatoes, they flower, so that might be a good time to take a photo. And we also know that tomatoes produce fruit, right? The tomato. And so that may be a time, and also it's ripening. Those are all good times to go out with the drone and take some images. And so following the flight, the data analysis will continue, and hopefully over time, we'll be able to find some trends that are caused by the root knot nematode from our gathered data. Real quickly before I finish, I would like to acknowledge people and groups. First of all, I'd like to say thank you to the USDA and NIFA. I'd also like to say thank you to Dr. Studiero and the funders of the Digital Agricultural Fellowship. Thank you to our collaborators, Chris Greer, the UC Cooperative Extension, San Luis Obispo County, and field trial collaborators, doctors Ollie Becker and Antoon Ploeg, Department of Nematology. Thank you to Nitran and the peer mentors who were extremely helpful during this RISE program. Thank you to Dr. Alexander Putman and the Putman Lab for all their support. And finally, thank you to my family for supporting me throughout the RISE program. Thank you all for listening. If you have any questions, feel free.